Hi children, good morning everyone. And in the previous class I told you about the components of the food, isn't it? Hope you are aware with all of them. Once we recall all of them and then proceed with our topic today. Okay, so what are the different components of food, children? They are carbohydrates, proteins, fats, vitamins, minerals along with water and fiber. They are very essential to maintain a balanced diet. So what is balanced diet? A diet which contains all the components like carbohydrates, proteins, fats, vitamins, minerals, water and fiber in proper proportions. Then it is called a balanced diet. Food should contain all these components in a proper level. Then only a diet can be said that it is maintained properly. Otherwise, you may suffer from various types of diseases. Okay, children. Now, let's start our lesson with test for carbohydrates. How to test whether the food substance contains carbohydrate. Okay, children. So, today's topic is test for carbohydrates. So, it's a simple test which we can easily identify the presence of carbohydrates. So, they are the starch that is present in a food. That is, I told you that the tubers, rice or paddy, sugar, jaggery, they contain the carbohydrates, more carbohydrates, isn't it? So how can we say that these things contain carbohydrates? Cereals contain carbohydrates, especially whole cereals. How can you say that they contain carbohydrates? So this we are going to test. For each test, you should know how to prepare the observation notes. What is it? First you should know what is your aim to do the experiment. So it can be experimentally proven. This is a test which can be experimentally proven in the laboratory. So for this aim, to test the presence of carbohydrates. What is your aim here? To test the presence of carbohydrates in a given food substance. In a given food substance. Next, what is it? Apparatus. That is materials required. Things that are required to do that experiment. That is called as apparatus. What do you need to do this test, carbohydrate test? Especially the chemical that is required is iodine solution. What do you need? Iodine solution. Iodine solution is required, and then you need two test tubes. Two test tubes or a petri dish, okay, or a porcelain dish you can say for that, and also a dropper and a substance that is to be tested, that is a potato. That is a tuber. You can take a grain. If it is a grain, you can finely powder it and take it. So now here I am taking a potato as an example. So any item that is the ingredient that is given to test can be taken in that place. Okay. Next, coming to the experiment. How do you do the experiment? In this experiment, first what you take? You take first that is the iodine solution. You are taking a test tube first. Take a test tube or potato. Take a item that is to be tested. That is, take potato, cut it into two halves. Two equal any halves or anything, or you can smash the potato or anything. If it is a powder, that is grains, you can powder them and take in a test tube or in a petri dish or in any manner you can take it. So before doing this, first you will take this and you will keep it ready. Then you have to dilute the iodine solution. How to dilute the iodine solution? You have to take 2 to 3 drops of iodine solution and put in one of the test tube, and then add 5 drops of water in it and dilute it. When it is not diluted, at first the iodine solution is dark brown in color. Then when you dilute it, it turns to light yellow in color. Okay children. So after that, you take 2 to 3 drops of dilute iodine solution and put on the cut potato. Then you can see, what do you see? So take a potato, cut it into 2 halves and take dilute iodine solution. So I told you how to prepare the dilute iodine solution. So you are taking the dilute iodine solution, whereby you can prepare it earlier, or while doing the experiment also. First you can prepare the dilute iodine solution, then you can take a potato and take 2 drops of dilute iodine solution and pour with a dropper over the potato. Okay children, then observation. What is your observation? What is your observation here? The potato, it turns dark blue or black. It turns dark blue or black. To which color does it turns? It turns dark blue or black. So from this observation you come to the result that the potato, if it turns dark blue or black, it confirms the presence of carbohydrate in the given substance. If a dilute iodine solution is added to the substance and if it turns dark blue or black, it confirms the presence of carbohydrates in a given substance. That is written as the result or inference. Okay, so if it turns dark blue or black, it is the test for carbohydrates. Is it clear children? So what is the test for carbohydrates? Test for carbohydrates, you have taken the dilute iodine solution and 2 test tubes you have taken. In one of the test tube you can prepare the dilute solution, iodine solution, and another test tube you can use it for the grain, that is any other grains or sugars, or also you can be tested for that. Or in a petri dish you can use the potato, or the grains also you can use in the petri dish. So in that manner you can use the test tubes and a drop or 2 drop for the dilute iodine solution. It is the test for the carbohydrates. So you can write down neatly about the test for carbohydrates in your notebooks after reading the textbook children. So from these inferences we can confirm that substance contains carbohydrates if the substance turns dark blue or black on addition of the dilute iodine solution. Then it is a confirmation test for the presence of carbohydrates in a given substance. Okay children, is it clear now? Okay, so the result is the substance contains carbohydrates. And what is the inference? Inference is if a substance turns dark blue or black on the addition of the dilute iodine solution, then it is a confirmation test for carbohydrates. That is the inference for this. Okay, so once again I repeat, what is the aim of this test? Test for carbohydrate. That is to test the presence of the starch or carbohydrates given in the given food sample or food substance, anything that is given to you. Then you have to take the materials that are required to prepare this experiment. First of all, you need dilute iodine solution to prepare this experiment. If you have the concentrated iodine solution, you have to dilute it by adding water to it so that it is diluted. Then you take one of the test tube, you take the dilute iodine solution, and with the help of the dropper in another test tube, if it is a grain powder, so then you can, in different test tubes you can take and do this experiment also. Or in a petri dish, there is a porcelain dish also. It is also called as porcelain dish. Petri dish is also called as porcelain dish. So in this also you can Take a potato and cut it into halves and pour 2 to 3 drops of dilute iodine solution over the cut potato. Then after a while you can see that it turns dark blue or black. If it is the case, then you can confirm that the substance, that is the potato, contains the carbohydrates. Okay, so here the potato is the that is the sample that is given to you, the food substance that is given to you, and it is asked to find whether it contains the carbohydrates or not. Yes, how you can say that the potato contains carbohydrates in them? Because when on addition of dilute iodine solution, it turned to dark blue or black. So you can say that it is a confirmation test for carbohydrates, presence of carbohydrates. Okay children, now I tell you about the test for proteins. Next is the test for proteins. So now you have to find whether the substance, how you can find it contains proteins or not. So how to find out? Mostly you know that the proteins are present in pulses, beans, peas, and in animal sources like milk, eggs, meat, and so on. So nuts and all also in plant sources, they also contain the proteins. So pulses, red gram, green gram, black gram, Bengal gram, all these also contains proteins. How to find out that whether they contain proteins or not? So we can say that these substances contain proteins by taking two chemicals. What are the two chemicals mainly? Copper sulfate solution and that is you can say that sodium hydroxide solution. Okay, so copper sulfate solution and sodium hydroxide solution are the two solutions that you have to take to make this protein test. So what is the aim now? As you have said earlier in the previous test, you have to write this also to test the presence of proteins in the given substance. Here the given substance may be any pulse or milk also you can take, egg also you can take and do the test. You can remove the egg yolk and you can do the test too for the egg also. And the apparatus required for this, that is material is milk. You are taking the milk and two chemicals as I said you earlier, they are copper sulfate solution, that is 2% copper sulfate solution you have to take. Copper sulfate, what is it? Yes, solution, and sodium hydroxide solution. Sodium hydroxide solution. You have to keep in mind these chemical terms. Okay, next 3 test tubes you have to take here. 3 test tubes you need. 3 droppers you need for this experiment. Okay children. So how much percent of copper sulfate means you need 2% copper sulfate solution and sodium hydroxide solution is about 10% sodium hydroxide solution. So what is 2% copper sulfate solution and what is 10% sodium hydroxide solution? 2% copper sulfate solution means 2 grams of copper sulfate in 100 ml of water. 2 grams of copper sulfate solution, 2 grams of copper sulfate, that is powder, in a powdered form, in 100 ml of water if you mix, it becomes 2% copper sulfate solution. 10 grams of sodium hydroxide, it will be in the form of pellets, that is like camphor, you can see this also. Sodium hydroxide, they are in the form of pellets and in 100 ml of water you can make a solution of it. That becomes a sodium hydroxide solution. It is copper sulfate is a bluish green in color, it is in a powdered form and you can make a solution by adding 2 grams of copper sulfate in 100 ml of water. You can prepare the copper sulfate solution, and then you have to prepare sodium hydroxide solution with 10 grams of sodium hydroxide pellets within 100 ml of water. If you take it and make a 10 grams of, that is 10% sodium hydroxide solution, you have to prepare in the first case, that is an experiment, doing this experiment or the method of preparation of this, how to do this. So here you are preparing this first, that is first 2% copper sulfate solution and 10% sodium hydroxide solution is prepared and kept. Then you have to start the experiment how to do. So what is an experiment here? You have to take 2 drops of copper sulfate in a sample of milk in a test tube. A sample of milk, you can say 5 drops of milk if you take in a test tube, and this is a test tube, children. This is a test tube. So you have taken the milk. This is the milk. You add the drops first, 2 drops of copper sulfate solution. Copper sulfate solution and 10 drops of sodium hydroxide solution. Sodium hydroxide solution. When you add this to the milk that you have taken as a sample. Now in the apparatus, in the experiment, you are taking the milk, isn't it? In one of the test tube, you are taking the milk, you are adding the prepared copper sulfate and sodium hydroxide solution from 2 grams of sodium, that is 2% sodium hydroxide, 2% copper sulfate solution. You are taking 2 drops of copper sulfate and adding with the help of the dropper, with the help of a dropper. Did you ever see the dropper, children? Hope you have seen this. This is a dropper. It will be like this. Hope you have seen in your laboratories. After reopening, you can easily see, and okay, with the help of the dropper, you can take it and 2 drops of copper sulfate you can add to this solution and 10 drops of sodium hydroxide solution you can add. Then it turns to violet or purple. So that is the observation. So after experiment, what you will do? You will see the observation. What is the observation? The solution turns violet or purple. Which color the solution turns? The solution turns violet or purple. So what is the result here? This indicates that the substance contains proteins. So what is the inference? If the substance turns violet or purple by the addition of 2% copper sulfate solution and 10% sodium hydroxide solution, then the substance contains proteins. So that is the inference. Okay, did you understand the test for proteins, children? So far you have understood the test for carbohydrates and also the test for proteins. In the test for carbohydrates, I said that you have taken dilute iodine solution. When you add dilute iodine solution to this sample substance, which it is, if it is a tuber, you can smash it or you can cut into 2 halves and add these drops over it. If it is a grain, you have to make it into fine powder and then paste it and then add this powder. Paste means in the form of, you mix a water in it and make a fine paste and then add the dilute iodine solution. Then you can find the result that it turns, whether it turns dark blue or black. If it turns dark blue or black, then only we can confirm the presence of carbohydrates in that particular substance. Now in the case of the test for proteins, you are taking the 2 chemicals, that is 2% copper sulfate solution and the other one is 10% sodium hydroxide solution. 2% copper sulfate solution means 2 grams of copper sulfate in 100 ml of water and 10% sodium hydroxide solution, which means 10 grams of sodium hydroxide pellets in 100 ml of water. You are preparing in the form of the solution and taking them to do the experiment. In doing the experiment, you have have taken the milk in a test tube, adding 2 drops of copper sulfate and solution and 10 drops of sodium hydroxide solution in this milk. Then if it turns dark, that is a violet or purple, then you can confirm the presence of proteins in that particular substance, that here we have taken the milk as the substance. Hope you have understood the result and difference, children. What is the result here? The substance contains proteins. What is the result? The substance contains proteins. Okay, and if it is about the, what I said about the carbohydrates, it turns to dark blue or black. Now you know what are the different substances which are mainly carbohydrate substance. They are tubers, all whole grains, and also the sugars, jaggeries and all, they contain the carbohydrates in them, that is the starch, presence of the starch. Okay children, and next about the proteins, pulses, beans, peas, eggs, milk, meat and all, they contains the proteins in them which are body building foods. They are very much essential for growing children like you people who grow continuously up to certain age. Till then you should take protein foods regularly. Protein foods also help you to the growth of the cells in your body continuously and make you healthy. Okay children, and next about the carbohydrates. If you take the carbohydrate foods, you get more and more energy. They are energy yielding foods. So carbohydrates and protein food is a must and should to take in your diet regularly to prevent various deficiency diseases. Protein energy malnutrition takes place in the children like you people if you do not take the required amount of the carbohydrate and protein level in your diets regularly. So it is a must for you to take them in your diet regularly. And coming to the fats, experiment to test the presence of fats in a given substance. Test for fats. How to test the fats? It's a very simple process children to test the fats. In testing the fats, you have to take a piece of paper, test for fats. You know that fats are present in oils mainly, isn't it? And also nuts. So for this example, you can take any pakodi or the oil foods, that is dosa, anything. If you take in a piece of paper, you can observe that after removing the food particle from the paper plate, then you can see the transparency of the plate can be seen over there, isn't it? That the paper turns translucent. That is translucent nature of the paper is seen, not transparent. It is translucent. Translucent nature is seen. So if you eat sometimes you eat for pakodi in a piece of paper. After eating it, when you see that paper, you can see that paper turn translucent, isn't it? Why that happens? Because that food contains the fat in them. You can identify with that the food contains the fat in them when the paper turns translucent. So it is the result, that is in the observation that you find when you take the fat foods in a piece of paper. So this also you can do as an experimental method by taking the groundnuts. Groundnuts you take and you smash the groundnuts over the piece of paper and you remove the groundnuts from them, and so after taking that is the smash over it, the paper also you can easily squeeze it. Okay, then you rub the groundnuts over them and remove the groundnuts, and you see the paper and put the paper against the light source and observe it carefully. You can see that the paper has turned translucent. From this experiment you can see that the substance that you have taken contains fat in them. Okay children, so this is a very simple test that you can identify easily whether the substance contains fats or not. Here what is the aim children? Aim is to test the presence of fats. What are the materials required to do the experiment? Groundnuts or any pakodi or any food material that contains fats, that is oil substance in it. You can take it, and a piece of paper is required to do this experiment. And how do you do the experiment? You take the groundnuts, you must squash, that is you what do? You make squeeze them the paper with the along with the groundnuts and you smash it. Then you remove the groundnuts and you put the piece of paper against the light source and observe, and you can see that what is your observation now? You can see that the paper turned translucent. So this confirms the presence of fats in the given food substance. Okay children, so that is about the confirmation test for the fats. That is, if we take a paper, if it turns into translucent nature, what is translucent nature? That the object is visible partially, not completely. It is not transparent nor opaque, it is translucent. So that is the translucent nature. So that the paper can be seen over it if it contains the fats in them. So this is about the test for carbohydrates, proteins and fats. Okay children, in the next class I'll tell you about the how to make a balanced diet even a cheaper manner, and what are the that is about the nutrition facts about the food materials. So different scientists who have discovered about the nutrition and so on. Okay children, so with this I end this today's class, and write down the homework that I give you today and come to the next class. So first one is write the confirmation test for fats in a given substance. Okay, that is, you can take groundnuts for instance. Examples, you can take anything. Now I am giving you groundnuts, you take it and write down. Next, second one, what are the chemicals required for confirming proteins in a given substance. Okay children, next, third one, given a substance turned dark blue or black on addition of dilute iodine solution. Why is it so? So this is the homework children. Complete these questions and come for the next class. Okay children, bye. Have a nice day.